My name is Sharon Donovan, and I'm a professor at the University of Illinois. And I do research in the area of pediatric nutrition with a focus on bioactives and their role in infant development. And today it's my pleasure to talk to you about Lactoferrin and Osteopontin, two bioactive milk proteins, and their role in neonatal intestinal development. This slide shows an overview of my presentation. We're going to begin with a discussion on the importance of postnatal gastrointestinal development. We will then move into talking about the role of milk bioactives. Why should we study them? How do we study them? And then we'll take a deeper dive into lactoferrin and osteopontin, and then we'll leave you with some take-home messages. So why study the gastrointestinal tract in the postnatal period? As you know, upon delivery, the infant abruptly transitions from predominantly receiving intravenous or parenteral nutrition via the umbilical cord to oral nutrition. And in response to this transition, the gastrointestinal tract undergoes significant growth and functional differentiation. And we know that this is influenced in part by genetics, by diet, and hormones. And while the gastrointestinal tract is doing this adaptation, we also have the development of the infant microbiome. Since infants are born with a relatively sterile gut, and it becomes colonized in that early postnatal period. And at the same time, the infant's mucosal immune system is now being presented with new dietary and microbial antigens, and it must be educated in order to tolerate these foreign antigens in order to avoid the development of food allergies or inflammatory bowel diseases. So if we look at these together, when we look at nutrition, we know that whether an infant is breast or formula-fed influences the intestinal structure and functional development, the microbiome development, and the mucosal and systemic immune development. But I want you to keep in mind as we go through that all of these are interdependent. And we know this from studies, for example, on germ-free animals who do not have a microbiome. We see abnormal development of the intestine as well as the immune system. And so all of these are dependent upon each other. However, today we're going to focus on the structural and functional development of the intestine. So human milk is really an incredible fluid. It's a complex living fluid, and while it provides all the nutrients needed for infant growth and development, what's really exciting are these bioactive or biologically active components. And it's thought that these are likely to contribute to many of the short and long-term benefits that are associated with breastfeeding. So these components include immune cells and cytokines, growth factors, the lipids, the milk fat globule membrane, human milk oligosaccharides, as well as microbes, what we call the human milk microbiome, and bioactive proteins. So future webinars will touch on other topics here, but today we're going to focus on two of those proteins, lactoferrin and osteopontin. So why do we study these bioactives and gut development? Again, much of the work comes from preclinical animal models, which show that when a neonate is suckled by their own mother versus one that's fed formula, that there's differences in their gut development. And specifically, mother-fed infants have increased intestinal mass, enzyme activity, absorptive capacity, and barrier function. So that tells us that there's likely components in that natural milk that are promoting the growth of the gut. We also know from preterm infants that those that are fed human milk, whether it's their own mother's milk or donor human milk, have a reduced risk of a disease called necrotizing enterocolitis, as well as late onset sepsis, and have improved feeding tolerance. Unfortunately, our studies in term infants are more limited because there's lack of non-invasive approaches for us to really test gut development. However, my laboratory, in collaboration with a colleague, have shown that you can use exfoliated intestinal epithelial cells that are shed into the stool to look at gene expression in the gut. And we've demonstrated, indeed, that the gene expression differs between those infants that are breast versus formula fed. And lastly, studies have also shown, linking back to immunology, that these specific milk components can improve not only intestinal development, but reduce intestinal inflammation. So all of these get us very interested in trying to determine what are the roles of these bioactive components, and ultimately, should we be adding some of these to infant formula? So how do we study this? And so we can go from what more simple models to more complex. And some of the most simple models are cell culture. So when you read the literature in this area, you see that some of these studies are done on cultured cells. And so the advantages is that you can test very controlled conditions. You can test different ingredients on different cell types, like gut cells or immune cells. You can really, though, only detect effects on that cell type. And we also can't appreciate, whether that component actually survives through digestion and is able to be presented to those cells. But it is good for understanding mechanisms. We can next move into our preclinical animal models. And again, these are more complex, but we can still control for the genetics and the environment, particularly with mice that are very inbred and they can be housed in similar situations and fed very similar diets. And we can use these animal models to not only test for safety of ingredients, but also their efficacy or effectiveness. And again, these are good for understanding mechanisms of action. And I'll talk later about some studies in rhesus monkeys and piglets. And then lastly, we can conduct clinical trials. So once the in vitro or tissue culture and preclinical animal models have shown the ingredients are safe and that they may have efficacy, we can move into human studies. Again, these are still done to demonstrate safety, and it allows us to capture more of the real life complexity of genetics and environment, since not all babies are inbred and genetically similar, and they live in many different environments. But as I mentioned, the limitation is that it's very difficult to get the samples, like intestinal samples or blood samples, even from infants, so that we can really test effects on the intestinal development. So now moving into our two proteins of choice. And so today we're going to focus on lactoferrin and osteopontin. And these are both proteins that we know quite a bit about and have been tested through all of these different levels of complexity. And in the case of lactoferrin, is already being supplemented to some infant formulas. So these proteins are between 75 and 80 kilodaltons. They are found in bovine milk, and the homology or the similarity between human and bovine is about 61 to 68%. We see the concentrations in human milk. So lactoferrin is the second highest whey protein. So it's a quite predominant protein in human milk. You can see that the concentration drops as we go from colostrum or that early milk to mature milk, but we're still in the gram per liter concentrations. Lactoferrin is also present in bovine milk, as I mentioned, but you can see the concentration is only about 1/10 that of human milk. But because it's present, we are able then to isolate that lactoferrin, and that's why we can use it as a commercial additive. When we think about its potential role in the intestine, there's other two questions that we have. One, is it resistant to digestion? So in order for it to exert a role in the gut, it needs to be able to survive in intact form. And this has been shown for lactoferrin, where about 4 to 9% of the lactoferrin in the milk that baby drinks can be detected in the stool in a completely intact form. We also know that there's receptors for lactoferrin throughout the gut, and that the lactoferrin in milk combine to those receptors. It is commercially available, and as I mentioned, it's already been added to some infant formula. In terms of osteopontin, we see a similar trend where it drops from colostrum to mature milk and is much lower in bovine milk. Keep in mind that this is a milligram per liter versus grams per liter, so the concentration is much lower in milk. Similar to lactoferrin, it's been shown to be resistant to digestion, as well as there are receptors present in the intestine. It is commercially available, but has not yet been routinely added to formulas. So turning to lactoferrin, before I talk about its role in intestinal development, I just wanted to mention some of its other actions. Many of these have been related to its potential roles in the immune system and in reducing infection. So a study in Peru showed that adding lactoferrin to oral rehydration solution could reduce the duration of diarrhea and increase the number of infants who are able to achieve a solid stool. So these were infants who were being hospitalized, recovering from diarrhea. Bovine lactoferrin in preterm infants has actually been shown in a number of clinical trials to reduce the incidence of late onset sepsis, so potentially having a role in the systemic immune system. Another study in Peru showed that feeding lactoferrin to children in that population reduced their colonization with Giardia and improved their growth. And then in a study done in my laboratory in piglets, we showed that feeding lactoferrin in a formula could reduce the severity of Staph aureus septicemia, which is a major cause of late onset sepsis in preterm infants. So many biological actions have been shown already for lactoferrin. In terms of intestinal development, a lot of this work has been from cell culture models. And just to mention, so lactoferrin, because it's very high concentration in human milk, we see that a lot of that lactoferrin is actually iron-free. Lactoferrin gets its name from lacto meaning milk and ferrin being iron. So one of its major roles is to bind iron. But 70 to 90% of the lactoferrin in human milk actually does not have iron bound to it. The rest, 10 to 30%, is binding iron, typically 1 to 2 molecules. And what's interesting about this is that studies in cell culture have shown that while both forms can be taken up into the cells and stimulate cell signaling pathways, this APO, or iron-free form, is more potent in its ability to stimulate cell signaling, and it could even move into the nucleus where it can add as a transcription factor and increase the expression of genes regulating cell growth. So these cell culture models suggested that lactoferrin might be playing a role in intestinal growth and development. So in my laboratory, we conducted a study where we were interested in looking at the role of lactoferrin added to infant formula on intestinal development in piglets. So we had a standard formula which contained some lactoferrin just in the bovine milk. Then we had 2 levels, 1 gram per liter or 3.6 grams per liter. So these are both within the range present in human milk. The piglets were fed these diets, and on 7 and 14 days, we obtained intestinal samples. And I'm going to mention, we wanted to look at proliferation, so we administered a compound that's similar to, looks like DNA. So when the intestine is proliferating or making new cells, this compound gets incorporated into it, and then we can detect it in those cells. So it can tell us how quickly the gut is proliferating or growing. And then we also looked at the villus height or crypt depth, what we call intestinal morphology, along with the numbers of these EDU labeled cells and gene expression. So we found that the piglets grew normally, and in most cases, they looked just like the piglets that were getting the control formula. But then when we started looking in the intestine, so I just wanted to show you a picture. So in the intestine, we have what we call villus, or villi and crypts. And on the villus is where the cells are actively digesting and absorbing nutrients. And in the crypts are where new cells are made. So when we administered this compound EDU, it was incorporated into these cells shown by these black dots. So we could see how many of these cells incorporated the EDU. And then those cells are made, they migrate up, and then they're lost from the tips of the villus. So when we looked at the number, when we looked just at this morphology, we could see that the piglets getting the 2 lactoferrin diets actually had deeper crypts than the piglets on the control diet. So there was no difference with villus height, but we found that the piglets getting the lactoferrin had deeper crypts. And so that's where these new cells are made. And other studies have shown that when you see enhanced growth, that these crypts become deeper. So then we looked at the incorporation of this EDU. And so we wanted to look more specifically if we could actually see differences in proliferation. So these show the age of the piglets, 7 and 14 days, and the different diets. And so you could see that in both the groups getting lactoferrin, we saw about a 60% increase in the numbers of cells that were proliferating. There didn't seem to be any difference with the dose of lactoferrin or with the age of piglets. So having the lactoferrin even at 1 gram per liter increased proliferation. And then we looked a little bit farther and we looked at a gene that we know from the literature regulates this process, this gene called beta-catenin. And we found that in the piglets on the LF3, or the 3.6 grams per liter, had a 3-fold higher expression. So these data suggest that lactoferrin maybe play a role in just regulating normal development of the intestine, but we think it may be particularly beneficial for those infants who have some compromised gut. So potentially for preterm infants or infants with intestinal injury. So maybe an infant that's had a diarrhea and needs to recover some of their gut. So it could have both normal development, but also potentially enhance the recovery from injury. So now turning to osteopontin, again, there was one study that I was part of where we conducted a clinical trial in China, and we had infants who were receiving either a standard formula with bovine osteopontin at half the concentration of bovine or human milk, or at the mean concentration of osteopontin in bovine milk, and a breastfed reference. These infants were fed for the first 6 months of life, and samples were obtained at various ages, at 1, 4, and 6 months. And again, most of the outcomes that were assessed were related to immunology. So we found that they grew similarly, but the infants getting the osteopontin had less fever, that they had lower levels of pro-inflammatory cytokines and higher level of IL-2, which is important in what we call Treg cells, which are actually reducing inflammation. So the combination of those two suggests that OPN is reducing inflammation and inflammatory markers in the serum of these infants. And we also found differences in that the infants in the higher OPN had higher proportion of T cells in the serum. So this suggested that OPN may favorably influence immune development and actually in some cases make them more similar to breastfed. So turning to the last few slides, this was a study where we looked specifically at bovine osteopontin and intestinal gene expression, and this was conducted using rhesus monkeys that were fed either formula with osteopontin at 125 milligrams per liter, or were breastfed. And so in this study, we collected jejunal tissue and analyzed the gene expression, and then we asked 2 questions. Are there differences in the pattern of gene expression between the groups? And we did this using a method called a weighted gene correlation network analysis, or WGCNA. And then what are the functions of those gene products? So what we found when we looked at the overall gene expression, when we compared the formula-fed monkeys with the breastfed monkeys, we found that there were about 1,000 genes that were differentially expressed in the jejunum or intestine of these monkeys. And most of those were down-regulated in the formula-fed versus breastfed. And the fold changes of these genes were pretty significant, from about -18 to +10-fold. In contrast, when we looked at the formula versus the formula with bovine osteopontin, we actually didn't see that many genes that were differentially expressed. But what was surprising, when we looked at those monkeys getting the bovine osteopontin versus the breastfed, we found that rather than there being 1,000 genes differentially expressed, there were only 217. So that was suggesting to us that the presence of the osteopontin was shifting the intestinal gene expression in the breastfed infants to be more similar, or the bovine osteopontin to be more similar to breastfed. So just to show you sort of the approach we take, so this is what we call these WGCNA modules. So what this does, when you're dealing with thousands of genes, you need to make some sense of it. So this program puts them into, clusters them into modules. And so you can see that in this case, the breastfed and the bovine OPN have higher gene expression than the piglet or the monkeys just on the formula. And so most of that, about half the genes fell into that module. And so just to show you, these are just how these different modules look. So these are this module 1, where expression was higher in breastfed, intermediate in osteopontin, and lower in formula fed. But you can see that there's different patterns of gene expression that we could detect. So we focused a bit on module 1, and then this was answering that question is what are they doing? So using a program called Metacore, we could assign functions to all of these genes, but we could also look, is there some common denominator? In this case, is there a common transcription factor that osteopontin may be working through? So we found that many of these genes were linked to this protein called CUX1. It's this gene, and this gene has been linked in other studies to stimulating expression of other genes and proteins that regulate cell cycle progression and cell migration. So we were basically kind of on the right track. And again, this paper is published if you would like to know more information, but the key take-home messages are that osteopontin at the level in human milk was able to shift overall gene expression of formula-fed monkeys with getting the osteopontin to be more similar to breastfed, and that we were able to identify several of these key transcription factors, and this helps provide insight into how osteopontin may actually be regulating gene expression in the gut, and then helps to provide potential targets for interventions. So I hope you have enjoyed the presentation, and here's your take-home messages that the early postnatal period is a critically important time for supporting intestinal development, and we know that if this doesn't go right, that infants can have long-term issues, things like food allergies, increased incidence of inflammatory bowel diseases, and other factors. We know that human milk is a complex biological fluid, and that these essential nutrients and bioactive components are likely playing a role in this sort of improved adaptation to postnatal life that we see with breastfed infants. Today, we focus specifically on just 2 of the proteins, Lactoferrin and Osteopontin, and I showed you some data that these have been shown in human studies to stimulate immune development, and also in animal models to affect gut development and gene expression. And really, these data help support this concept that the addition of key bioactives to infant formulas may help us to narrow the gap, these developmental differences between breastfed and formula-fed infants. So thank you very much.
Thank you very much, Sharon.
I see I'm about 3 minutes over. I hope that's okay.
That's perfect. So Sharon, moving on to the first question.
Okay.
Is there evidence that lactoferrin and osteopontin could be acting synergistically?
Yeah, that's a great question, and unfortunately, I didn't have time to present that today. But there are a couple cell culture studies that this hasn't been shown yet in animal models, but in cell culture, first of all, people showed that because of the structures of these proteins, that they can actually bind together. And this is a sort of a, chemical interactions between them, and that this is quite potent, so they can stay together. And when the lactoferrin osteopontin complex was presented to cells, by the same group, by Bo Lauderdale's group, they actually found that this complex could be more potent than the components individually. And, that brings up another really, I think, important take-home message is that, human milk is a combination of many, many, many ingredients. And oftentimes when we study these in human clinical trials, it's, we're just adding one ingredient to an infant formula. But I think that's a fantastic point that we know that many of these ingredients may work synergistically, and some may even have, opposite effects. That if something is really promoting growth, maybe another component is coming and, putting a little bit of brakes on that because you don't want to necessarily have uncontrolled growth. So while my last point is that we can add these ingredients, we can show they're functional, we also need to consider that, adding simply one ingredient is not going to be the end all, and it's certainly not going to represent what a breastfed infant observes when consuming human milk.
Great. Then what would you consider being the main differences between lactoferrin and osteopontin in terms of their benefits in infants?
Yeah, so that's also a really good question. We've known, lactoferrin has been studied for a much longer time than osteopontin, and so we know a bit more about it. And some studies have suggested that it may impact iron absorption or iron status that hasn't always been consistently shown. So that would be a function that osteopontin doesn't have. Lactoferrin, as I've shown, can have antibacterial, antiviral effects as well, and that hasn't been shown as much for osteopontin, where osteopontin has really been shown to have effects. And so osteo meaning bone and pontin sort of stimulating. What osteopontin was actually first identified in bone, and that's how it got its name. But other studies have shown that it can really impact the immune system, and particularly the T cells in the immune system. So this is why, when we presented the human study with the osteopontin, we were focused on the immune system. So, I think OPN is considered more classically immunological, but our monkey study really showed that it can also exert roles in the intestine and affect gene expression. And other studies have shown it can act as a transcription factor, so stimulating gene expression. But again, a lot of that had been limited to looking at immune cells. So while we are seeing some overlap in their functions, they do still have distinct activities, and in some cases, may actually be maybe complementary to provide, them together, because they can potentially provide, additional benefit having both of them there.